Thanks, Natasha. And I would like to thank Natasha, Ellen, and Angela for the invite and for preparing this nice setup, and to you, everybody that is attending. I'll try to give you guys some flavor of what SmartCella is. I'll try to squeeze in 15 minutes. Let's see if I can make it. SmartCella is a Swedish biotech company. We're located just across the street here in Alfred Nobel's Alley. And our business is pretty much segmented in two areas, targeted delivery and regenerative medicines. On the targeted delivery, we've developed endovascular catheter called the extraducer. It has been invented by Dr. Staffan Holming, our Chief Medical Officer at Karolinska Institute. We have a cell therapies, within the regenerative medicines, we have a cell therapies branch, which I'm head of, and we have two products. At the moment, derived from pluripotent stem cells. I'll talk a bit about this, and a modified cell therapies branch located in Solna, where we have early research and focusing on mRNA-based therapies. Regarding the extraducer, one of the main challenges in delivering cell and gene therapies, but also not only but also small molecules and other biologics, is the right place, the right time, and the right dose. Most of these are delivered systemically, where a couple of challenges are faced regarding toxicity, insufficient doses, and accumulation mostly in the liver. We have developed the extraducer, which is an endovascular device that uses the Seldinger principle. We use pretty much the vasculature of the patient to navigate with the catheter and reach the organs that are targeted using imaging technologies, and then a needle goes outside from the vessel, from the blood vessel, and direct into the organ of interest. This allows for not only a reduction of the cost of the surgery, because some of these surgeries are done at a very invasive fashion, like open chest, and with using the catheter, we reduce the costs of patient handling. The patient pretty much go to the hospital and have the delivery of the payload at the same day, and leaves even on the same day. I'll give you a brief imaging. I'll fast forward this, but just for you to have an idea how the endovascular, how the extraducer works. Just wanted to show. It's a very thin device of 1.5 meter length, and we use imaging to navigate the catheter in the patient's vasculature, and once we reach the organ, then the needle pops out, and the payload is injected. This is just for you to see the thickness of the needle. It's pretty much a hair wide. This is an example of an application for cardiac, so for delivery of payloads in the heart. We have currently our own program, but we also are collaborating with other. Yeah, the sound is not clear.
Extraducer is a minimally invasive device designed for safe, targeted delivery of therapeutics to heart tissue. Conventional endovascular equipment is used to navigate a guide catheter into the ventricle. The extraducer, which consists of a 33-gauge infusion needle positioned inside an outer protective sheath, is advanced through the guide. Throughout the procedure, alignment of the radiopaque markers on the needle and the sheath is verified under fluoroscopy. At the target site, the extraducer is advanced from the guide catheter under fluoroscopic guidance. After releasing the hemostatic valve, the needle is advanced from the sheath, penetrating the ventricle wall until the marker, which acts as a depth-limiting collar, reaches the endocardium. Securing the hemostatic valve locks the needle's position prior to payload injection into the myocardium. After injection, the hemostatic valve is released, and the needle is retracted into the sheath until the markers align again. The device can now be repositioned to the next injection site or removed from the body. The The extraducer is designed with a small, flexible needle to maintain its position in the beating heart despite contractions. This minimizes tissue trauma while enabling precise payload delivery across multiple injection sites and may enhance tissue payload retention. For more information, please contact SmartCella by email.
So you can see there's a lot of potential applications of this device, and we can deliver pretty much any payload into several organs. We're currently focusing on oncology and cardiac and CNS applications, but the potential for using it in other organs is quite outstanding. Our pipeline, so we have some internal programs, but also partners working with us. And we are currently using the extraducer in a phase 2B trial in the US with the company Xylocor for delivery of an AAV therapeutics in the heart for refractory angina. We're planning to submit excuse me, a clinical trial application here in Sweden for the use of gemcitabine for pancreatic cancer for targeted delivery of this chemical at KI hospital in Solna. And internally, so we have the Smart 1, 2, and 3 programs, and I'll try to give a flavor on these internal programs. But before that, so the facility that I mentioned, so just across the street, we have capabilities in terms of process development, analytical development, and GMP manufacturing. We have 2 clean rooms of 60 square meters each. You're more than welcome to chat with me after and plan for a visit. And our business model is mostly to take projects that have the potential of becoming a best-in-class therapeutic, and with a previous analysis of health economic and market access, and making sure that there's room in the field for those programs, bring those programs to phase 1 to A trials, and then out-license these to large pharma. We have pretty much a team of, so it's a multidisciplinary area, as you guys know. So we have a team of process engineers, biophysics, stem cell biologists, and bioinformaticians. And we also have, all the technical operation functions that are required to bring or to translate these therapeutics into human application, like regulatory affairs, quality assurance, and quality control. We also have a QC lab for release of these therapies. And our foundation is focused on structure, so and make sure that these, when we develop manufacturing processes, these processes are developed not only phase appropriate but also risk appropriate. We want to develop processes that are amenable actually to be used and scalable to support later phase trials. Also risk, our goal is to develop what we call predictive manufacturing, where we use novel or sensors to understand and to monitor better the microenvironment of these cells during expansion and differentiation, and understand if the process will fail, let's say in 2 days or 3 days or the next day. Understand that and or predict that, and use control strategies so that we don't lose a batch and it's still successful, and we don't waste money. And also the development, when developing a process, we focus on increasing the yield by process intensification strategies, and reduce the cost of goods and the labor requirements, because these processes, as you know, they depend a lot. They're currently very manual and open, and they depend a lot in operator judgments. So the operators have to be highly skilled in these processes. Just a brief. There should be okay. On the right side, you see the cardiac cells, more mature cardiac cells that we are using. So our Smart 1 program, we use progenitor cells, so our cells are still not beating, which we believe that is an advantage. We use the extraducer to deliver these cells directly into the myocardium. And this targeted delivery, as I mentioned at the start, not only allows to ensure that actually the cells go to the right place, but also reduce the cost of patient handling in the hospital. Because we're using progenitor cells and single cells, we deliver these cells as single cells. We believe that has the possibility to reduce the doses that are currently employed or used by other developers. And also because our product are progenitor cells, so the length or the duration of the manufacturing process is shorter, which also reduces the cost in terms of using GMP facilities. Our target target in terms of process development and analytical development with our IPS platform is to be able to develop processes that are more efficient, and will be able to reduce labor by 40% and materials costs by the same value, and increase the throughput of the manufacturing. As I said in the beginning, so we deliver with these cells with the extraducer, so we reduce it's a way less invasive procedure than open chest procedure. Our second product in the pipeline, so SmartO2, it's a technology or a protocol developed by Johan Eriksson at Karolinska Institute. We have licensed this technology. These are dopaminergic neural progenitor cells derived or differentiated from IPS. And just to give you a flavor of the potential of this technology, on the top left, we see, so this is a rat experiment in terms of efficacy. This is a striatum, and the cells, our progenitor cells, are delivered in one side of the putamen that has been removed of dopaminergic neurons. In the orange, in the area highlighted by a yellow dashed line, it's the size of the graft of our cells. And the competitors, or one example of a competitor, actually the area of the engraft is 15 times bigger than ours. But what's interesting, and it's in the middle column, is to understand the efficacy of these cells. And just trying to quickly help you reading the plot if you don't know already. The point where efficacy is achieved is when the line, the plot line, crosses the horizontal dash line. So in our case, in our SmartO2 program, and using a Parkinsonian rat model, we achieved efficacy 3 months after injection of the cells, whereas competitors actually achieved it 7 months by 7 months, so 4 months later, which suggests that actually our SmartO2 product has a faster efficacy compared to the competitors. And on the right is histochemistry staining of the engrafts. And for you to have an idea, so green is staining for dopaminergic cells, and our grafts are extremely rich in terms of these cell types, and showing a high purity, where competitors have not only few of target cells, but also several contaminants. To terminate, just a brief on our SmartO3 program, which is related with the modified cell therapies segment. So we use IMSCs as a vehicle to deliver mRNA. We have a proprietary protocol to differentiate IPS into IMSCs. We have IP on that, and our initial program on this segment is delivery of mRNA for treatment of osteoarthritis. And we did a combination or an analysis of factors that are involved in the process of cartilage regeneration. We did studies in vitro, and we have selected 2 factors. Here are called A and B, but in this case, what showed better performance is the factor B. And we're using a rat model, and what this plot shows is the comparison of the osteoarthritis model, a common treatment, which is hyaluronic acid, MSCs, which are also used to treat this type of indications, and our IMSCs with one factor that we call factor B at high concentration and low concentration. What we see in this plot is that actually there's using our technology that promotes cartilage regeneration until at least 7 weeks. We are currently doing a long-term study for 6 and 9 months. I can share that the data for 6 months is quite promising, and corroborates these results. And you can see in the histology images that actually there's true degeneration of the cartilage. We also have a model, although it's a bit, it can be argued, a model of pain relief with these rats, and also promotes our technology, our IMSCs with these mRNAs reduce the pain in the rats. Last but not least, we are a team of the executive management team has several. It's quite international team. All the SmartCella, and also we have a strong team in the board, in our board. But who is in the trenches and make this a reality are actually the guys who are in the labs, and some of them are here, and all kudos to them. So happy to answer any questions that you have, and thank you.